My name is Kimberly Skeet, and my work is focused on studying how our blood evolves as we age and develop disease. I am particularly interested in the evolution of a small population of cells known as hematopoietic stem cells, which continuously replenish our entire mature blood pool. As we age, our hematopoietic stem cell populations acquire somatic mutations which can confer a functional advantage known as driving mutations, causing one cell or group of cells to contribute disproportionately more to the mature blood pool. This phenomenon has been termed age-related clonal hematopoiesis, or ART, and has been found to increase an individual's risk of progressing to cancers such as acute myeloid leukemia or AML by up to tenfold. However, these same driving mutations are commonly observed in elderly individuals who do not progress to disease, and the question remains, why do some people with these mutations progress to disease and others remain healthy? In particular, I'm interested in how passenger mutations might play a role in shaping how our blood cells evolve towards cancer. By that I mean, we often study cancer evolution through a lens of positive selection. We are interested in those driving mutations and how they rise in frequency in our blood population. However, we know that the majority of somatic mutations are going to be mildly damaging and removed through a process of negative selection. And so we are asking, does our blood evolve according to the positive-only model of evolution that is frequently explored in cancer research? Does it fit a model where we only have negative selection acting on passenger mutations? Does it fit a combination model where the interplay of selective pressures might stall clonal expansions? Or does it just evolve neutrally? Finally, how do these patterns change prior to cancer development? To model the evolutionary processes in blood, we have generated over 5 million simulated blood populations evolving at a neutral, negative, positive, or a combination of selective pressures. We then condense each population down to a concise set of summary statistics and train a deep learning model to use those summary statistics to distinguish between those 4 different classes of evolution. When we test our model using a held-out set of simulated data, we find that we are able to predict the correct evolutionary class around 86% of the time. After achieving a high accuracy in discriminating between evolutionary classes in artificial data, we apply our classifier to identify the evolutionary patterns and targeted sequencing data from blood of around 300 healthy individuals and 100 individuals who subsequently progressed to AML. Right away, we find that the majority of our individuals do not fit the neutral or positive-only classes of evolution, and we find signatures of negative selection either acting alone or in combination with positive selection in over 60% of our samples, regardless of a later cancer diagnosis. Further, we find that many genes are commonly mutated across these different evolutionary classes, but we do observe a small number of genes which are exclusively mutated in negative and neutral models as well as positive or neutral models of evolution, suggesting that mutations in these genes might play a large role in shaping these patterns of evolution. Finally, we ask if negative selection acting on passenger mutations is indeed associated with longer AML-free survival. We performed a survival analysis and found that individuals fitting negative and neutral classes of evolution remained AML-free for longer than those with signatures of positive selection. Our work finds that we cannot study cancer through a lens of positive selection alone, and that passenger mutations play an important role in shaping clonal dynamics of progression to cancer. Thank you to both the Awudella and Morris Labs, our funders and co-participants.